All right, thank you for the introduction and the opportunity to give this talk here this morning. So I'm going to focus on an application of some work that I've done using the MassTech AP MALDI source. And in this case, doing sequential analysis from the same tissue section, looking at different classes of molecules in this study. So what we were looking at here is in situ imaging of an acetylglucosamine. So this is an O-linked sugar, which has been underrepresented in the literature. So we had come up, so I'll take you through the process of how we developed the method to do this analysis and how it fit into the sequential analysis from tissue sections. So just start out with some rationale behind why we might want to do sequential imaging. Typically, diagnostic biopsies are quite small. They may be on the order of only 1 or 2 millimeters in the case of pinch biopsies, non-invasive diagnostics. And because these are so small and they might be squished and distorted, sometimes the histological diagnosis can be challenging, and other specialized tests are needed, but only a few molecules can be used on each section with these specialized tests like immunohistochemistry. And so of course, if these biopsies are that small and are being used to diagnose, there's not a lot of material left over for research, so we need to be able to do as much as possible from a single section of tissue. And as well, if you're using serial sections, you don't necessarily have the same cells in the serial sections, and the alignment of serial sections can be challenging. Now, as of course all of you know, mass spec imaging allows us to detect hundreds to thousands of analytes from a single tissue section, so we can get a lot more information from one section of tissue. And if we carefully plan our experiment, we can do multiple mass spectrometry images from the same tissue section. So getting into this O-glycnac protein modification, this is a single sugar co- and post-translational modification that occurs on serine and threonine residues. It's reversible, it's been implicated in a lot of different biological functions, including stress response, epigenetic regulation, proteostasis, and in my interest area, it's also highly involved in tumorigenesis. So there are 2 enzymes involved, one that puts the sugar on, O-glycnac transferase, and one that removes the sugar, O-glycnac hydrolase, which I'm going to refer to as OGA through the rest of this talk. So we wanted to come up with a method to analyze where these O-glycnac modifications were located on proteins in tissue. So here is our methodology. We used a rabbit liver tumor model, the VX2 model. So we had tumors that were grown for about 9 to 11 days, harvested and snap frozen. We sectioned at 12 micron thickness. All of our major Cs and enzymes were applied using an HTX M5 sprayer. We had digital images of serial sections for histology annotation using a Hamamatsu scanner. And we did all of the imaging using a AP MALDI source on a thermofusion Lumos instrument. And in this case, because this was a method development study, we didn't go to the super high resolution. This was all done at 100 microns. But one of the things I want to point out that was a challenge in doing this is we needed a really high humidity incubation for the O-glycnac hydrolase. So we have what we refer to as, if I can get the mouse over here, we call this our spa treatment. So it's a piece of white ball on the bottom and 2 rolled up chem wipes that act as little pillows, your little spa pillows. All of that saturated with water, the slide sits on top of it, elevated in that steamy environment in the oven. But what we saw when we did these long incubations overnight at 37 degrees in this very high humidity, we had weather patterns. And the moisture would condense on the lid and it would rain down on the tissue, and you would end up with something that looked like this. I like to say the tissue had measles. And so what we found, borrowing from the DNA and RNA world, that if you heat the top of the dish, so this is a reptile heating pad. Reptiles are very prominent in mass spec imaging. We use reptile sand in sublimation devices. I use the reptile heating pad here. But what that did was keep the lid the warmest part of the incubation chamber, and it prevented that raining from happening that caused artifacts in the imaging. So first we wanted to start out with a standard and figure out our limit of detection. And so to do this, we did a dilution series that we spotted both on bare glass and also onto some tissue that we didn't add any enzyme to. And then we imaged these spots to figure out what our limits of detection were. Ultimately, we had about a 37 nanogram per gram limit of detection on tissue. A little bit different calculations than what I was used to. All of my background had been in more pulsed MALDI with distinct pixels. So a little bit different calculations in determining the limit of detection because our line scans didn't ablate all of the matrix. So we had our laser at this point focused at about 33 microns and our line spacing was 100 microns. So we had to take that into account that we're only sampling a third of the area when we were calculating limit of detection. Next, we looked at how well this worked on standards. So we used a standard glycopeptide with one O-GLICNAC modification and treated with different concentrations of OGA to figure out what was going to be optimum. And so here, just to briefly go through these results, we have looking at what should be the intact peptide, and in all 4 spots where we treated it with different concentrations of OGA, we don't detect the intact peptide. But if we look at either untreated or using an inactive mutant form of OGA, we do see the peptide with the modification. If we image for just the sugar, as shown in the green in the center panel, we see it, in this case, we're seeing the sodium adducts of N-acetylglucosamine in all 4 spots. The 0.25 mg per mil didn't look super great there, but we don't see the O-GLICNAC in the untreated or the inactive. We're maybe getting a little bit of laser-induced fragmentation. And then finally, we can look at the peptide minus the modification, and we see it readily in all 4 spots that are treated, and we believe that the standard was not 100% glycosylated, so we do see some unmodified, or some peptide without the modification in ones that weren't treated. So moving on, we were able to do this on tissue. So here is imaging of O-GLICNAC in 3 different rabbit liver tumors. And so we're able to see very good signal and localization to the tumor area in all 3 of these samples. Most abundant in viable tumor. Most of these tumors had some necrotic areas, so we weren't seeing the O-GLICNAC as much in the areas of necrosis. We do see a little bit of signal corresponding to the mass of the O-GLICNAC, and this on the control tissue, and this is likely due to a little bit of laser-induced fragmentation. And in this case, we were looking at the water loss species that is sodiated. That tended to be our best detected species on tissue. So now we also wanted to be able to correlate what we saw with these O-GLICNAC modifications in tissue to what are the proteins that they might be coming from. So to accomplish this, we had sections of tissue. We wanted to treat things as similarly as possible. And so we did an on-tissue digestion using little punched out pieces of SDS page gel that we loaded with trypsin and placed them on specific areas of interest on the tissue. So we could target the tumor, we could target necrosis, we could target normal liver. And after 4 hours of incubation and digestion, the peptides that were digested were transferred into the gel. They migrated into the gel as the digestion happened, extracted the peptides and did nano LCM-SMS to identify some of the proteins that carried this O-GLICNAC modification. So a few of them are shown here in the table of some of the peptides that were detected with the modification. And interestingly, we were able to, in the imaging experiments, and I'll show you more detail of this in a few slides, see at least one of the proteins that carried an O-GLICNAC modification in our imaging experiment. So we found that histone H2B had a modification site, and we also were able to image a peptide from histone H2B that showed similar localization to where the GLICNAC was in the tissue. But again, the GLICNAC is going to be coming from all of these different proteins, and we can't differentiate how much comes from each one. So to do our sequential enzymatic treatment, this was the workflow that we used. We found in the method development that if we didn't first cleave off the end-linked glycans that are big and bulky, we didn't have as good access of the OGA to the GLICNAC residues, which are much smaller on the proteins. So our first step was to take the frozen sections, do a carnoise fluid wash to remove a lot of the lipids and small molecules, treated with PNGASF, sprayed with CHCA as a matrix, and then imaged the end-linked glycans. And then took that section, washed off the matrix using ethanol and carnoise fluid, treated with OGA, so now to release the O-GLICNAC in situ, again used CHCA matrix and imaged that. And then finally we washed that off, treated with trypsin to do analysis of peptides in situ, CHCA matrix, and MALDI imaging. So looking at the results, we were able to detect well over 100 glycans in the tissue with several different spatial localizations, either to normal tissue, viable areas, necrotic areas, etc. So a wide variety of different end-linked glycans that were detected. Not shown on this slide, interestingly, we also detected peaks corresponding to glycogen in the normal liver. They were insanely high, and when I talked to my collaborator, she said, yeah, the animal care person felt bad for these rabbits and was feeding them marshmallows. So not surprisingly, there was a lot of glycogen stored in their livers. But it was just an interesting finding, so never know what you're going to find in your imaging experiments. So then we did the O-GLICNAC imaging on the same section, and so here we can see the signal corresponding to that area of viable tumor around the edges and not as much in the necrotic area in the center. Again, this is the water loss sodiated species. And then finally, the tryptic peptide imaging. So even after 2 rounds of enzymatic treatment on these tissues, we're still able to get robust peptide signal by tryptic digestion. And we were able to compare, some things that co-localized or differentially localized versus some of the other either end-linked glycans or the O-GLICNAC. So now we've done these 3 separate imaging data sets. We have our end-linked glycans from the PNGF treatment, the OGA treated for the O-GLICNAC, and the tryptic digest. But these are all from the same section, so we want to be able to correlate everything. And so one way to do that is, well, we can just slide them together, but how accurate is that, and you can't easily see how things compare. But one of the neat features with the Skills Lab software is if you have an optical picture in common, which was that picture of the slide without any matrix on it, you can co-register all 3 data sets or more. I've done up to 8 data sets all co-registered to each other. And now we can visualize them as if all of these distinct data sets were collected at the same time. So what I'm showing here is in green is the O-GLICNAC signal, in blue is one of the end-linked glycans that we detected, and in red is a tryptic peptide of hemoglobin from the final analysis of tryptic digestion. And so we can now further mine these data and see what's co- or differentially localized compared to each other. And so then just briefly, I also want to just give a teaser for my poster today. That's also what some of the data was collected on the mass tech source with the thermofusion Lumos instrument. And in this case, we wanted to look again at sequential imaging, but now combining the small molecule analysis with a technique called MALDI immunohistochemistry. And so this is, some of you have probably heard about this. This is antibodies that are produced by Ambergen that have a photocleavable peptide reporter tag on them. And so we wanted to correlate what we were seeing in the small molecule imaging, trying to target a drug species with some of these known protein marker changes. So first we did metabolite imaging using DHV matrix positive mode at 50 micron resolution. Unfortunately on this system, I did not readily detect the drug that it was dosed with. So interestingly, when you come to my poster, I did this analysis on the AP MALDI source, which is atmospheric pressure. I did this also on a Bruker-Tinstoff instrument, which is intermediate pressure, and I used a Rapiflex, which is high vacuum. And surprisingly, I had best signal for the drug on the Rapiflex. And so I think there's just some variability of how well things ionize depending on what type of vacuum in the source. But we did detect a number of small molecule metabolites, lipids, and lysolipids. So shown here, and for those of you who can't read it, we have lysine in blue, spermine in green, and a lyso-PC in red in these 3 different samples. And the 3 samples are a control. These are, again, those same rabbit livers that I showed you that we did the other study with, used for this study as well. And so we have a control sample that was untreated. These are treated with drug-eluting beads to deliver the drug to the tumors. And so this sample is vehicle. These are beads that have no drug in them. And then in the middle, they were treated with drug that were beads that were loaded with the imiquimod drug. And so after I did the small molecule imaging, we washed off the matrix and treated with a panel of antibodies and imaged for the peptide markers after UV cleavage. And so this is just using the standard Ambergen IHC staining protocol, use CHCA matrix, and this was also imaged at 50 micron spatial resolution. So showing here 3 of the antibodies from the panel. Beta-actin is in green, HMGB1 is in red, and pan-CK is in blue. And so we can see, good localization and some correlation of localization of some of these proteins with different features in the samples. And then again, we can co-register since these are collected from the same samples. I just focused on the one that was drug treated. And so here we're combining the spermine signal in green, HMGB1 in blue, and pan-CK in red. And so we can visualize this as if it was all collected at the same time, despite them being very different sample preps. So in conclusion, I hope you appreciate the new method that we've developed for the imaging of O-GLICNAC in tissue samples. I've shown you 2 different sequential imaging workflows, one where we did 3 steps, then linked glycans, O-GLICNAC, and tryptic peptides. And then in this other one, 2 different modalities of drug and metabolite imaging, followed by MALDI IHC. This sequential imaging of the same tissue section allows us to get a lot more information from a lot less material, and the co-registration allows us to discover some of these biological interactions that we might miss otherwise. So I'd like to thank all of my collaborators who worked from several different institutions, so myself at the University of Texas, my collaborators at Simon Fraser University who developed the OGA enzyme, the samples from MD Anderson, Ambergen for supplying the enzymes, Boston Scientific and CPRIT for funding, and MassTech for giving me this opportunity to speak here today. I'd also like to mention, for those of you who want to learn more about mass spec imaging, I hold an annual short course and workshop in my lab. MassTech is one of the sponsors of this workshop. So if you're interested or know somebody who wants to learn more, it's 2 days of classroom instruction, and you also get to pick 1 day to come to the lab and do the entire process from sectioning through data analysis. So that'll be the week of July 15th, if anybody's interested. Also, just for those of you who want to see more imaging data, there will be an imaging workshop in Madison at the end of July. Registration is open. Abstracts are due on Monday. So would love to see some of you attend that. And then finally, there'll be the International Mass Spectrometry Imaging Society meeting in Munster in September. And abstracts are open for that as well. So with that, I'm happy to take any questions.
Hey, Eric, excellent work. So my question is, your tissue after with glycochemistry, trypsin digestion, and laser irradiation, so is it a little bit beat up at the end?
It is, yeah. So especially with the line scan, and a little bit harder to tune the number of shots because there is a minimum rep rate that you can run on the source, and we have to time everything to the speed of the Orbitrap. So yeah, you do, if you stained that section afterwards, and you can see it in that picture I showed where we did the limit of detection, you can definitely see some marks. Even with an irregular pulsed laser where you can be a lot more judicious with your laser power, when you do that many analyses, especially on frozen tissue, you do see some damage. I see a lot less damage on fixed tissue.
What was the number of post-step? 122?
244.
240. Thank you.
You're welcome.
Thank you so much for such a fabulous talk. So kind of following up on that question, could you adjust like the thickness of the tissue at all to adjust if you wanted to have higher lateral resolution? Like how would you adjust your method so that you can have higher resolution?
Oh, we certainly can have higher resolution. As Jill indicated, this source can go down to 5 microns. Mm-hmm. My issue was, in this case, with as much as we were trying to dig in and accumulate and the speed of the Orbitrap, these 3 tissues, each run through took 32 hours. So it was a 64-hour experiment to do it at 50 microns. And of course, in the way of ASMS, I'm collecting my data like the day before I have to get the poster printed. So in that case, I was a little bit limited, but certainly we can do higher resolution and we wouldn't need thicker tissue to do that.
Sounds great. Excellent work. Thank you.
Thanks.
Any more questions?
Hi, really cool talk.
Thanks.
So your standard was a peptide with a single glucanate on it.
Right.
When using this as a standard, or a reference rather, how can that fully suffice as a reference when, especially in a digested sample, you could have peptides with more than one O-glucanacylation on it?
So we were using that as a reference for doing the method development of figuring out parameters like how much enzyme to apply, how long to do the incubation time, because this was an enzyme that had never been used on tissue before. So it was just a starting point of something that was cheap and easy to use, required a lot less sample prep than dealing with tissue sections, and would be quicker to do the analysis on. And then there was still a little trial and error. We had what we thought was best on the standard, and then we still had to tweak it a little bit when we got to tissue.
If we have no more questions, let us start.